I'm going to pull out one of the myofibrils. See, I'm going to pull this out. See that? See what I've done? I've pulled this one out so that you can see it. You know, a myofibril is a long linear sequence of sarcomeres. So each of these, 1, 2, 3, 4, 5, seen, many more, not seen, each one of those little units is called a sarcomere. And the best visual impression I can give you is that if you were sitting at a railroad crossing and there was a long train of tank cars coming along and going across your crossing, you'd say tank car, tank car, tank car, tank car, tank car, right? Mm-hmm. And they'd be joined at a coupler, right? Well, there's your tank cars. 1, 2, 3, 4, 5, 6. And they're joined at a coupling, and the coupling is called the Z-line. So each one of these black lines there is a Z-line. Z-line, 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 Z-line. You understand?
Mm-hmm.
You follow me?
Yeah.
Okay? Now that's basic information up to this point. Now this might not seem, for some, it might not seem to be very significant, but it's profoundly significant. All right? And what I'm trying to get you to realize is the depth at which you are thinking critically, which is what's going to save your tiny heinies in nursing school. If you can answer this. I'm curious to know if anyone can. You got to go back to chapter 2. You got to go back to chapter 3. And if you start putting all those together, you can answer this question. But that's not like taking a standard question and asking, how many legs does a dog have? You have to think. You'll notice that in my cell right here and up here also, this cell is multinucleate. You also need to remember that a muscle cell is about 90% protein. It should stir up a thought. Then my question is, you know the answer if you'll think, why must a muscle cell be multinucleate? You get that when you are really good.
For like the production of RNA or something? For production of RNA or something like that?
It's close.
For the production of protein.
How? Why?
Or transcription.
Transcription, translation, right? It's 90% protein, right?
Yep.
Remember, you're constantly repairing and tearing it down and building it back up.
Especially in Culpepper's class.
So, for you to make protein, you make it through transcription, translation, and where is all the information for making protein held?
Ribosome.
In the nucleus, in the DNA. See? So muscles, wherever you have a cell with largely protein, you've got to have a lot of DNA to go through that process. Now, not that that's going to win you any award, but it does tell you one thing, and if you figured that out, you are way ahead of the majority of the people that will be in that school over there. Because you're able to think critically. And in your patient care, they're not all cookie-cutter patients. There's all kinds of complications. There's all kinds of thought processes that you'll have to decipher and come up with. And that's the main goal that I have this semester. Probably the top 3 goals I have this semester is to get my students to begin to think critically, think out of the box. You do that, you really really have done yourself a big favor. I'll promise you that. All right. Now what I want to do is to show you that what is the majority of a sarcomere? What is it made up of? And how am I able to do that? See that? How am I able to do that? We're going to go into great depth on that today. First off, you got to remember that all these myofibrils, thousands of them in a muscle cell, they all are made up of these sarcomeres laid end to end. What separates one sarcomere from another sarcomere are the Z lines, which are proteins. It's a protein, another kind of protein. They act as couplers between these units. The sarcomere is the basic unit of contraction of a cell. In other words, when I flex my forearm, the reason I'm able to do that is because my muscles shorten. The basic unit of contraction in that shortening would be each of the thousands upon thousands of sarcomeres. All right. Now, in a sarcomere, you will have two kinds of myofilaments, or myofibers. These are the thick myosin, which is in the middle right here, the orange. And notice they've got these little bitty, what they call myosin heads, or some books call them cross bridges. These are the myosin filaments. There. All right, see? Got it?
Mm-hmm.
Those are thick protein, big guys with little finger-like things like this on the surface. All right? Here, originating at the Z lines and coming in and very carefully geometrically situated between the myosin is another protein called actin.
That
So
sounds
you got to remember
familiar.
the thin filaments are actin, the thick are myosin. Myosin have the cross bridges. Got it? All right. You have another protein that's called chitin, which attaches to the myosin, and these are involved in the relaxation process of muscle tissue. In other words, when I contract my muscles and I shorten it, I flex my forearm, then if I let them relax, all those proteins are going back to their original shape. And one of the factors in that will be the chitin protein. All right? Now, let me see if I can show you here. This is just another picture of the same thing. There. Same thing. But it shows you how the myosin, here's the sarcomere from Z line to Z line. Remember, there's all kinds of them in a row. Here are the thin actin myofilaments. Here are the thick myosin myofilaments. And there are, here in this case, they're calling them cross bridges. But they can be also, I believe your book calls them myosin heads. All right? Now, what happens when they're stimulated, the following is kind of like what it's going to happen. All right? In this case, I'm going to be the big fat myosin, okay? I am the thick myosin myofilaments. Here are my little cross bridges, see? Myosin heads. When the muscle is relaxed, these are the nice and cute actin filaments. See, right here, these are actin. These are actins, and this is actin. You see? I'm the big fat ones. I'm the myosin. So, normally what happens, I do not come in contact with them. See? But when stimulated, and if calcium is going to be the stimulator, what happens, there's a change in the physiology of the action.